Hi friends, today we're going to see the past year NEET questions for the chapter Molecular Basis of Inheritance, and today in this video we are going to complete part 5. So let's begin with the first question, which was asked in the year 2004. In a mutational event when adenine is replaced by guanine, it is a case of dash. So what does adenine have to base pair with? In transition substitution, a purine is replaced with another purine base, adenine with guanine or vice versa. In a transversion substitution, a purine is replaced with pyrimidine base or vice versa. Frame shift mutation is a type of mutation which or where the reading of codons is changed due to the insertion or the deletion of nucleotides, while transition is the formation of RNA over the template of DNA. And when adenine is replaced with guanine, where you have both purines, therefore the right answer will be transition, which is option C. Next question. The telomeres of eukaryotic chromosomes consist of short sequences of dash. So telomeres are sticky terminal ends of the chromosomes. It has a heterochromatin and a repetitive DNA. So you have a diagram to your right side which clearly tells you telomeres are repetitive nucleotide sequences at each end of the chromosomes, that is in the terminal ends, and they are sticky in nature. So the telomeres of the eukaryotic organisms will be rich in adenine, which is option C. Adenine rich repeats. DNA replication of a bacterial chromosome DNA synthesis starts from a replication origin site, and this question was asked in the year 2004. So when we talk about this, let's discuss a few points about DNA replication. What do you mean by DNA replication? How can you describe it? How can you even define it? See, DNA replication, when it comes, so of course when we study DNA replication, it comes under molecular biology. DNA replication is a biological process of producing two identical replicas of DNA from the original DNA molecule. And DNA replication occurs in all living organisms, acting as the basis for their biological inheritance. So this is something that I would like to add on for DNA replication in common. Now they're asking about the replication of a bacterial chromosome DNA synthesis. It starts from the replication origin site, and it moves in the bidirectional way. Replication begins at the site called as ORI site, and this is the origin of replication site, and it produces or precedes on both sides of the origin of the replication site, that is ORI site. Unidirectional replication is very rare when it comes to the RNA primers, and that is supposed to be involved in both prokaryotes and eukaryotes. So therefore the right answer is option D. Next question. During translation initiation in prokaryotes, a GTP molecule is needed in dash, and this question was asked in the year 2003. So what happens here? For the formation of the initiation complex during translation of messenger RNA, GTP is also required. The initiator is so this you will already know because we saw that codon, right? AUG is the initiator there, and this codes for the formyl methionine in prokaryotes. So this is actually an association of the 30S mRNA with formyl methionine tRNA. Therefore the right answer is option D. Next question. Which one of the following triplet codes is correctly matched with its specificity for an amino acid in protein synthesis or as a start or stop codon? So we all know that AUG is the start codon or the initiating codon, and UCG codes for serine, triple U, that is uracil, codes for phenylalanine, UGC codes for cysteine. So now they have asked for which one of the following triplet codes is correctly matched with the amino acid. The one which is correctly matched is option A, UAC tyrosine. Next question. What would happen if a gene encoding a polypeptide of 50 amino acids, 25th codon UAU, is mutated to UAA? So mutation is called as sudden change or defined as sudden change. UAA is actually a stop codon, while AUG is a start or an initiation codon. UAA is a stop codon actually. Therefore, at the 25th amino acid, the synthesis of polypeptide will stop. If UAU is mutated to UAA, the process will come to an halt because it's a stop codon. Therefore, it will stop the reaction. Therefore, the right answer to this question is option B. A polypeptide of 24 amino acids will be formed. So it's irregular, right? It's not complete. Next one. In the genetic code dictionary, how many codons are used to code for all the 20 essential amino acids? Very easy question. What is the answer here? Out of the total 64 codons, 3 codons do not make any sense. So it's totally 61. Hence, only 61 codons are used for the formation of the 20 essential amino acids, while the other 3, they do not make any sense. I mean, they don't code for anything, or they don't have a purpose. They're just there. Therefore, for 20 essential amino acids, number of codons is 61. Next question. Degeneration of a genetic code is attributed to dash. So before you go into the answer, when we talk about degeneracy of codons, is the reductancy of the genetic code, exhibited as a multiplicity of the 3 base pair codon combinations that specify an amino acid. Now this degeneracy of genetic code is what accounts for the existence of synonymous mutations. So you can ask me why is a genetic code degenerate? There is sometimes, so there are more codon combinations. There are more codon combinations than there are amino acids. The genetic code is degenerate because more than one codon sequence can code for more, actually they can code for the same amino acid. Codons that specify for the same amino acid are called as synonymous. So degeneration of the genetic code is attributed to the third member of the codon, which is option A. So according to the Warble hypothesis, transferred tRNA anticodon has the ability to warble its 5'N by pairing with an even non-complementary base of an mRNA codon. So it corresponds to the third base degeneracy of the codons. Next, out of 64 codons, 61 codons code for 20 types of amino acids, and they are called as, this is also easy question, they are called as degeneracy of genetic code, which is option A. You can ask me what about the rest of the 3? They don't make any sense. They don't have a purpose, or they don't have any work to do. So out of 64, it's only the 61 codons which code for the 20 essential amino acids. The 3 successive nucleotides of the bases code only for one amino acid. Wobbling refers to the third base degeneracy. So out of the 64 codons, 61 codons code for 20 types of amino acids, and that is collectively called as degeneracy of genetic code. Next question, types of RNA polymerase required in nucleus for RNA synthesis. Any idea what is it or how many? You require 3. Okay, to your side, I'm going to give you an explanation to that. You have RNA polymerase 1, 2, and 3, right? You have 1, 2, and 3. So 1, if you see, it is found in the nucleus and responsible for rRNA synthesis, while 2 is responsible for mRNA synthesis, and the last 3 is responsible for tRNA and 5S RNA synthesis. So each has a specific action to do. Next question here, gene and cistron words are sometimes used synonymously because they are dash. Why they used like that? Because cistron is that segment of DNA which specifies synthesis of polypeptide. That is why we synonymously use. Therefore, one gene contains one cistron. So they synonymously use both. The right answer is option C. Next question, mutation generally produces dash. So what did I say? What is mutation? They are sudden changes. They are sudden changes, right? So what comes here? They might not be favorable. So they actually called as lethal genes. While if you take polygenes is a gene that is single dominant allele of which expresses only a unit of trait, while lethal genes or on expression stop producing vital products. You must have seen in the same part where we spoke about the UAA code, which is a stop codon. Instead of reducing to 25 amino acids, it produces 24 amino acids, so it gets a halt. So mostly mutation, they sometimes stop the production of the vital products that are required for the organism. Therefore they are called as lethal genes. Therefore the answer is option D. Next question, one function of telomeres in a chromosome is to dash. So what did we see in part 3? We told that telomeres are repetitive units of nucleotides. So one function of the telomere in chromosome is to dash. So if you see telomeres, they occur in the ends, right? And they are sticky in nature. So they are in the terminal ends, they are sticky in nature, and they are rich in adenine. Now you should be able to answer. Since they are present in the ends, they seal the ends of the chromosome. Seal the ends of the chromosome is a function of this. The telomere is a ribonucleoprotein complex composed of protein component and an RNA primer sequence which acts to protect the terminal ends of the chromosome. Therefore, this prevents chromosomal disintegration and prevents the ends of the chromosome from being processed as a double-strand DNA break. Therefore, the right answer is option C, seal the ends of the chromosome. Got the answer? With this, we complete the part 5 of molecular inheritance chapter. Hope to meet you in the next part with another set of questions and explanations. Thank you.